Good afternoon, everyone, and thank you for joining us today. I'm Katie Stubbs, and I'm from Alzheimer's Research UK, who, along with the Alzheimer's Society, fund Brains for Dementia research.
Prynhaen da pawb, diolch am ymuno â ni heddiw. Fy enw i yw Rachel, a dwi'n dod i ti heddi o ganolfan Cardiff y prosiect Ymennydd ar gyfer ymchwil dementia. Byddaf yma i gyfieithu unrhyw cwestiynau sy'n dod trwy Gymraeg. My name is Rachel, and I work at the Cardiff Centre for Brains for Dementia Research, and I will be present to translate any questions that we receive in Welsh today.
It's our pleasure to welcome you here today for the second event in our series. The purpose of these events is to share the ongoing work of Brains for Dementia research, and also to thank you because none of it would be possible without you, our study participants, partners, and your families. We're recording this event and the others in the series so that you can watch them back or catch up if you're unable to make any of them. Today, you'll be hearing from Dr. Rebecca Sims, who leads the Cardiff Centre in the BDR network, and from Dr. Megan Torval, a researcher based in Cardiff. You are welcome to submit questions at any point during the talks. To do so, you just need to find the Q&A button at the bottom of your screen, and another box will appear where you can type your questions. You're welcome to submit questions in Welsh, and when it comes to the Q&A sessions, Rachel and I will be back on screen and be putting the questions to Rebecca and Megan.
Mae croeso i chi gyflwyno cwestiynau ar unrhyw adeg yn ystod y sgwrsiau. I wneud hynny, cliciwch ar y botwm Q&A ar waelod eich sgrin a bydd blwch arall yn ymddangos lle gallwch chi deipio eich cwestiynau. Mae croeso i chi gyflwyno cwestiynau yn Gymraeg, a phan ddaw at y sesiynau holi ac ateb, bydd Katie a fi yn ofyn cwestiynau i Rebecca and Megan.
So without further ado, it's my pleasure to introduce our first speaker, who's Dr. Rebecca Sims, who leads the Centre in Cardiff in the BDR network. And she'll be talking about her research, which is looking to identify risk genes for Alzheimer's disease. So over to you, Rebecca.
Thank you very much. Sorry, thank you very much. I think I unmuted at the same time as I got unmuted. I'm just trying to find the presentation that obviously was open. There we go. So thank you all for joining us today. As Katie said, I lead the BDR cohort centre in Cardiff. I'm going to talk to you a bit today about some of the work that the group does finding risk genes for Alzheimer's disease, which includes your kind contributions to BDR. So this project is the European Alzheimer's Disease Biobank, which is, as the name suggests, a European collaboration. It was initiated in 2016 and funded through an EU Joint Programme for Neurodegenerative Research, JPND grant, and includes 15 European countries, of which obviously the UK is one. And the project essentially aims to identify some of what we term the misinheritability of Alzheimer's disease. So we know that Alzheimer's disease has a genetic contribution, estimated to be between 56 and 79% of disease, and that this heritability is a statistic that estimates how much of disease is due to genetics. The work we've done over previous years has identified risk genes for disease, but there's still a lot of heritability, genetic heritability for disease that is not currently accounted for by the known risk genes. And actually, we think that approximately 50% of this disease heritability is still unknown. And EADB was formed to try and identify some of this missing heritability. So why are we specifically interested in identifying risk genes? A lot of the work we've done over previous years have highlighted specific genes for investigation and further research, but it's also highlighted biological functions, so biological pathways that we know are dysfunctional in disease. And this allows us to model disease in various systems, animal models, cell models, and helps us identify targets, not just necessarily the gene itself, but maybe genes and proteins up or downstream of the gene that potentially would be good targets for therapeutic intervention. And identifying these risk genes and pathways also allows us to develop risk scores, which potentially looks at identifying people with higher genetic risk of going on to develop disease. And this can be potentially for use to identify people that would be good for specific clinical trials, but also in the long term would potentially help us to have more person-specific medication. So the EAD project has assessed over 21 million variants across the genome. So what this project has done is taken what we call a microarray, which is a small chip, which normally I'd have one to show you, but unfortunately I'm not, as you can tell, I'm not in the lab at the moment, so I don't have one to hand. But it's essentially a very small array, which is probably about 3 centimeters by 3 centimeters, which contains millions of variants of known genetic variation across the genome. And what the project has done is used over 20,000 new individuals with Alzheimer's disease and compared them to over 22,000 elderly people without disease. And then through collaboration with other existing cohorts, has increased the number of people to essentially perform what we call a case control analysis. I should mention that the BDR cohort, a number of BDR participants are in this sort of initial EADB stage here. And what we then do is use what's called a case control design to basically look at which genetic variation is seen more often in people with disease compared to people without disease, and test that for statistical significance with regards to the difference of the numbers in cases and controls, as we term it. So the data from this is now published online if anybody is particularly interested. This is the paper that is currently on a resource called medRxiv, which is a preprint service for health sciences. What this means is that the paper hasn't yet been through peer review process, so it hasn't been assessed by other experts in the field, but it is available for people to look at, and it's currently under review for Nature, the journal Nature. So this is the results of the study, which I will describe. I know looks a bit scary. What this is called is a Manhattan plot, and essentially every little dot, so every blue or dark blue dot you see on that, and also the red dots, is a genetic variant, what we call a SNP, a single nucleotide polymorphism. So this is a single base change in the genome, and the 21 million that I just mentioned earlier. So there's 21 million dots on this picture. The dots correspond then to the association signal, so the statistical difference that we see between those people with disease and those people without disease in our cohort. And where we see a statistical difference, you see the line gets taller. I'm not sure whether you can see my arrow on here, but essentially where these lines come right up, this is statistically significant difference. They've got to get over this red line here. If they get over this red line, we deem that to be a genome-wide association. So essentially we correct for the number of tests we've done, and it's statistical at that level. So you can see that there's a lot of genes here that we're identifying, some known. So we've got ApoE as the sort of longest well-established risk factor for late onset Alzheimer's disease, and a number of other genes that we've seen previously. So complement receptor 1, which Megan may go on to talk about later, system as well, and all the red ones you see here are all the new ones that were identified by this particular study. Sorry, I can't move forwards for some reason. There we are. So what we've also done with regards to this, sorry, that all went the wrong way around, but what we've also done with regards to this study is develop new risk score predictions to predict disease, so what we term polygenic risk scores. It's designed to test whether the polygenes, so therefore multiple genes or multiple variants of smaller effect, conferring an aggregate risk for disease onset. And these generated scores can then predict those that have disease or will go on to develop disease, importantly. So we can predict those that are likely to go on to develop disease in a different data set to the one that we generated the scores from in the first place. And this is a very bit complicated figure, but I will try and work my way through and explain it. So what this does is use these risk scores, so we're identifying people that we think are most likely to go on to have disease, and saying whether we can identify conversion from normal cognition to any dementia. So the polygenic risk score we've developed from this data set, does that tell us if people are going to convert from having normal cognition to having dementia? And we see, so these are different, on the left here are just different data sets that we've used, different cohorts that have been used for this. And what we see here is that these studies, so these 4 studies that are across the other side of this line, we can predict with statistical significance those that are going to go on to develop some form of dementia with the risk score that we've developed. This is the same thing so a risk score, but going from normal cognition to Alzheimer's disease specifically. And we can see here that we do slightly better because more or less all the cohorts fall the correct side of the line. So we can pretty much, we've got some certainty of predicting who goes on to develop Alzheimer's from having normal cognition. And then also, quite importantly, looking at those individuals with mild cognitive impairment. So around 50% of people with mild cognitive impairment go on to develop full-blown dementia, either standard dementia or Alzheimer's dementia. And it's quite important for us and clinical utility to be able to identify which those people that have mild cognitive impairment will actually go on to develop dementia. So this is looking at using the prediction model again with regards to genetic risk profiling, which ones will go on to develop any dementia. Again, these are just different cohorts that have been used. These are cohorts that have individuals with mild cognitive impairment and longitudinal data to say whether they've gone on to develop disease. And then again, from mild cognitive impairment to Alzheimer's specifically, and our test is a little bit better in this cohort. So new genes equal new research. So this paper is not formally published yet, so it's only, we finalised this the end of last year, and it's already been utilised to regards to identifying genes for new research. So a lot of the work is going on in Cardiff with the Dementia Research Institute, so looking at animal models of some of the risk genes, also looking at some of the functional genomics, so looking at the pathways that they're involved in and looking at the networks of genes, and also going on to look at complex cellular models as well, which I believe Megan will probably talk to you a little bit about in her talk. So this is where the sort of genetics that we're identifying is leading, and hopefully at some point will lead to a therapeutic intervention target as well. So future work with regards to the consortium and what we've been doing with the ADB. So sorry, I just realised I haven't changed the name on this. Oops, I will go back. So EADB is formally finished now. The funding has ended, and it's going on to a new project which has just been renamed this morning to Rapid EU, and I haven't changed it on this slide, so apologies. But what this project is going to aim to do is develop not only a genetic risk profile that I've discussed in this presentation, but also look to integrate other forms of genetic risk and environmental risk, and try and develop a more comprehensive risk score for dementia that has clinical utility. So it'll be looking at the sort of shared genetic risk with other traits. So we know that people who've had type 2 diabetes or in earlier life and cardiovascular problems are at a higher risk of going on to develop dementia. It's also going to look at integrating blood marker data, so where we see increases in certain proteins in your blood with people living with dementia and Alzheimer's. As I said, cardiovascular risk factors, and also more environmental and social type risk factors with regards to mood or social isolation, and trying to look across all these areas to develop a more comprehensive risk model that could potentially predict those at highest risk of going on to develop dementia later in life. And that's a very quick whirlwind, and I'd be very happy to take any questions anybody may have, and I thank you all for your attention.
Thank you very much, Rebecca, for your talk. We'll wait and see if anybody has some questions coming through, but I had just a few questions I thought I'd like to find out about. So you mentioned there about these scores that your work is helping to develop. Do you have any idea of, it's in the research stage at the moment, but when that might be something that people could be offered in clinics to understand, their risk of potentially going on to develop dementia symptoms?
So there are a few risk sort profiles already on the market, as it were, not necessarily clinically used, but on the market with regards to sort of 23andMe and other sort of large commercial. There are other commercials entities that do these types of tests as well, that do give you a sort of risk profile score. The main issue with them at the moment is that while we can identify those, especially those at highest genetic risk of going on to develop disease, they're not as sensitive or specific as you would need for a clinical test. So that's the idea really with Rapid EU, is to try and add in some more risk factors, as it were, not just the genetics, but other risk factors to develop a more robust test that would have clinical utility. So it's edging that way. And I think part of the utility of these type of risk profile scores, it will actually be more for the precision medicine type approach. So we do see people having different types of genetic risk for disease. So some people have dysfunction in immune pathways, some people have dysfunction in protein trafficking pathways. So actually, if we can identify which people are developed disease through which mechanism, that could potentially help with therapeutic intervention for those people.
Interesting. That's really, yeah, good to know. We've had a couple of questions come through now. We mentioned one of the big project you've been working on had some EU funding, and so it's a question about how is leaving the EU affecting the research and cooperation with some of your European partners.
Yeah, unfortunately, it is affecting us at the moment. So the collaboration is still definitely strong and working well. So part of Rapid EU is that it is going for more European funding, and at this stage we are not included in that European funding. The UK is not taking part in that round. For most European funding rounds going forwards, I believe we will be included. There are certain things we're going to be excluded from, but the majority of the collaborative research we will in the future be included in. I think just this next sort of 1 or 2 years, it will affect us purely, I think, because obviously there was a lot of uncertainty around Brexit and when it would happen and what would exactly happen in that. So it is affecting us at the moment. I think we are going to have a lull with regards to the funding over the next couple of years coming through EU systems, but hopefully the collaborations are strong and it will push through that for the next sort of funding rounds in the next 18 to 24 months.
Good. So one of the other questions we've had is about, so I don't know if you'll know the answer, do you know if there's any particular blood type that has a higher risk factor? Is there any link between how genetics determines kind of blood group and then for dementia risk as well?
No. So obviously blood group, obviously you need specific genes to identify your blood group, and there are arrays that you could, like genetic arrays where you can look at blood type in correlation with risk. There's been nothing that links specific blood types to disease risk though. That's obviously not the case for other complex diseases, but for dementia there's been nothing robustly associated.
So one of the other questions we've had is about the work you're doing. Is it data? So when you're identifying these genes, are these typically in people who are alive and have given samples, or is this a mix of both, living participants and also people who've passed away that you have samples from?
It's a mix of both. So with regards to BDR itself, BDR contributed to the sort of new data that was generated as part of the project, and we had DNA from living participants that had provided blood samples, and also from the brain banks where the DNA was taken from brain tissue. So it is both, and it's the same for some of our collaborators as well. It will come from brain banks as well as from living participants.
And I've got a question. The next one is about data sharing. So it asks, is your genomics data available for other researchers?
So it will be. It's in the process of going through publication at the moment. So the summary statistics, which essentially just gives you a list of the variation that we've looked at and a statistical p-value, what we call a p-value, to look at statistical significance of that. They will be published and publicly available. The remaining data, so the sort of individual level data, is anonymized, so you can't identify any people from the data set. But that will be available through a request type process.
Great, thank you. And then I think the last question we have at the moment is one about how, I guess it's asking about how different types of research work together. So the question says, I noticed you were using animal research. Is it possible for people signed up for brain donation to specify they don't want their brain to use in a project using animals? So I guess this touches on some of the ways in which research teams use different types of experiments to get their answers. I wonder if you've got a comment on that.
So the BDR participants won't specifically be used in any animal projects. So the the actual samples from those participants are used to identify the genes of interest. They're not used then in the animal modelling system. There are essentially, what happens in the animal models is that the genes of interest are modified in the animals to see if we can see differences in the phenotypes of those animals, then whether they have sleep disturbances or eat differently, that type of thing. So none of the actual human samples will ever, in our centre at least, is used in animal modelling, and that would be, if it ever would be, that would be part of the consent process. So people should rest assured that if they haven't signed up for that, they won't be used in that way.
Fab, thank you. Well, thank you so much for answering those questions. I think we're ready to move on to the next speaker. So thank you, Rebecca. And if there's any more questions for Rebecca, we can always bring her back at the end to answer those then. So now it's my pleasure to introduce Dr. Megan Torval, who is also based in Cardiff, and she's going to talk about her research that looks at aspects of the immune system and what she's learning about how the immune system and dementia are related to each other. So over to you, Megan.
Okay, thank you for that introduction. And thank you to the organisers for today's event for inviting me to speak. So yes, my name is Megan Torval and I'm a postdoc working at Cardiff University, and I'm interested in dementia. And I'm going to talk to you today about my work in the lab to understand what our immune system, what role it plays in dementia. And my slide doesn't want to move on. Oh, there we go. Before I launch into the talk, I just want to give you a very quick introduction to me and a couple of other people, because it helps me to introduce you to the topic. So I grew up in Devon, maybe you can hear that in my accent, I'm not sure. And I went to Imperial College London, where I studied biochemistry as my undergraduate degree. And that is where I first became interested in neuroscience. But like a lot of people my age, when I finished my undergraduate degree, I didn't have the next part of my career lined up. So I went back home to Devon and I lived with my parents for a year. And I'm really glad that I did that, because I was able to spend time with my grandma Betty, who you can see here. And grandma was living with dementia. And we spent a lot of time together that year and it was really nice. But she used to get these urinary infections, which would make her really confused, and it was quite distressing. And she was experiencing what I now know to be delirium. And it was around about this time during that year that one of my professors from Imperial College sent me an advert for a PhD up in Edinburgh, which was studying that phenomenon. The combination of an infection and underlying dementia. And I'll talk a little bit about those themes in a little while, but I just want to introduce one last person before I start. And that is my, come on mouse, there we go, Grandma Joan, whose birthday is today. She's 85 today. And we chat a lot. And we chat a bit about some of the work that I do. And I phoned her up one day, not that long ago, and she said, well, I want to do something useful as well. So I've been to my GP and I've just signed up to be a Brains for Dementia researcher participant myself. So that's really cool, and no pressure from me on that front. So she does all the cognitive testing and she's agreed to donate her brain at the end as well. So that's cool. So my first point, what is dementia? So here you can see on the right hand side we have a cognitively normal, healthy looking brain, and on the left hand side is the brain from somebody who lived with dementia. So dementia is not a normal part of aging. It is a chronic and slow burning disorder, and it's caused by brain disease or injury. And people who live with Alzheimer's disease or any type of dementia experience memory disorders, personality changes, and impaired reasoning. The question that I am asked most often when I tell people that I do dementia research is, what's the difference between Alzheimer's disease and dementia? And the answer is that dementia is an umbrella term which refers to a whole host of different diseases, and Alzheimer's disease is one of those diseases. So Alzheimer's disease is a type of dementia. So diagnosing different types of dementia is really, really tricky business. The clinicians will usually do lots of different cognitive tests to try and work out which parts of the brain are affected, and then they will attribute it, the dementia to a specific disease. But that's not always clear cut, and it's sometimes not until post-mortem that a neurologist can look at a brain section on a slide and ascertain precisely what disease it is. So here we have various different disease tissues, and a common hallmark of different types of dementia is the accumulation of proteins. So here in Alzheimer's disease, we can see accumulation of amyloid protein, which forms plaques. And there are also tau tangles in Alzheimer's disease. In Parkinson's disease, there's accumulation of alpha-synuclein. In Huntington's disease, there's accumulation of huntingtin. CJD is like mad cow disease, but in humans, and there's accumulation of prion protein. And lastly, in ALS, which is what Stephen Hawking had, there's SOD1 protein. So there's a huge part of dementia research which focuses on these proteins. They exist in a normal, healthy brain. They just don't exist in this way all stuck together like this. So we want to understand, what are these proteins normally doing that they're now not doing, and what caused them to all stick together and be like this, and can we get rid of them? And if we can get rid of them, then does that even help? We don't really know. And the reason this is important is that the buildup of proteins throughout the brain affects the surrounding cells. This is a picture of a neuron. So the neurons are the electrically excitable cells of your brain. So they're the guys that do all of the heavy lifting, really, that allows you to think and feel and move. And these cells in dementia, they have these, so the cells are all connected in really intricate networks, and they're connected by synapses that kind of, I'm doing this gesture, I don't know if you can see me in the top corner, but the cells meet and have these junctions. And during dementia, the buildup of these proteins, these junctions die back. And so these synapses die back kind of like a flower when it's wilting away. And we want to know whether we can prevent that dying back of the neurons, dying back of the synapses. So what has the immune system got to do with that? Because normally we think of the immune system in terms of when we're sick. So, you know, like when you've got a cold or a flu. The immune system is the first line of defense in your body. So there are cells which patrol around in your blood, or they sit in all of your tissue and organs, and they keep a lookout, first of all, for baddies. So that could be a virus or a bacteria or any sort of infection. And once they've recognized a threat, they send out little chemical messengers that say, come here to immune cells, other immune cells, and they say, come here to the site of infection, there's something going on and I need backup. And once those cells arrive, the other immune cells, I'm going to show you this little video. This is an immune cell chasing a little bug around, and it eventually engulfs the bug, and then it eats it, and it's got enzymes and really acidic conditions inside, and it can kill that little bug. So it can either do that, or it can release chemicals that punch through the cell and they burst the cell open and kill it. Or it could, if it's a cell from us, one of our cells that's infected, it will do the same thing. And then more cells like this one on the right-hand side will come up and clear up all the mess. But a really important part of any immune response is conflict resolution. Once we've dealt with the threat, it's all gone. We need to then settle everything down, because if these immune cells keep going, then they'll cause loads of damage to our own cells. So there's lots of signals that are released that tell everyone to just calm down, clear everything away and go back to normal. So what has that got to do with dementia? Well, in the brain, there are these cells that you can see here. They're called microglia. And you can see they're very motile, they're wriggling around. And these guys are the resident immune cells in the brain, and they are constantly on the lookout for any invading things or anything that's just not quite right. And you can see in this next slide, these cells are responding to some damage caused by a laser beam in the middle, and they're putting out their projections, and those projections will kind of feel around, figure out what's going on, and remove any debris and clear up the mess. And then hopefully things go back to normal in a normal, healthy brain. So in Alzheimer's disease, you'll remember I said that we have the buildup of plaques and tangles. And here this is essentially like painting by numbers, a technique that I use all the time in the lab. And in red here, we've got amyloid plaque staining. And the microglia normally look like over on this side of the image, where they're kind of evenly spread throughout the tissue, and they're kind of just doing their own thing. They've got loads of arms and legs sticking out all over the place. But when there's a plaque, they all start to gather around the plaque and try and fix it and see if they can help out. And like that video I showed you, these cells are capable of eating stuff. So they clear up what we call debris or the amyloid. But in Alzheimer's disease, the proteins that are accumulating, the amyloid and the tau protein, are produced and accumulate at a rate that is quicker than these cells can do anything about. So these cells basically become overwhelmed. It's like they're bailing water from a sinking ship. They're completely overstimulated, and they just start eating things willy-nilly with no regard for what they're eating. And we think that that might be how the neurons and the synapses start to die back and get injured. So this is the area of research that I work on. If we can understand these processes, then maybe we could find a way to dampen down the immune response from these microglial cells and get everything back under control, and potentially improve the course of the disease. So we know that the genetic studies point us in the right direction, which genes or which gene products, the proteins, we need to be looking at. And we know that they affect Alzheimer's risk. But once we've got this post-mortem brain tissue, then we can understand where the proteins are in the Alzheimer's brain compared within a healthy brain. And that can inform what might be going wrong in the Alzheimer's brain. And in addition, understanding these things can help us to develop tests with which we can diagnose Alzheimer's disease, and also drugs to treat Alzheimer's disease, which we all know are sadly lacking at the moment. So I'm going to leave it there for now, because that's where I am with my research at the moment. So I'm afraid I don't have much more exciting data on that, but I'm happy to take questions now if anyone's got any. I'm sorry if I whizzed through that so quickly.
Thank you so much for that, Megan. So we've had a couple of questions come through. Somebody's thanked you very much for your brilliant explanation of your research area, and they want to ask what you know about FTD or Pick's disease, and is there any kind of research around that you would recommend people look at?
So I did my PhD on a bit. So FTD is a difficult disease because it's there's a spectrum of diseases that have different hallmarks. And I did some research on like one specific part. You know, when I showed all the different proteins in all of the different diseases, one of the proteins that is affected in FTD is tau protein. So it's got the tau without the amyloid protein. I'm trying to think what research would be kind of accessible. I'm happy to have email back and forth with people afterwards, because I can't think of any off the top of my head that's super accessible, but I am happy to answer questions about it. Sorry, that wasn't very useful.
No, I think that's fine. I know that we have some research contacts in Cambridge who focus quite heavily on FTD. So actually, Barbara, who asked this question, if you want to give us an email after this event, then we can kind of point you in the right direction towards some of those. A similar sort of question about vascular dementia. So how does your work and your interest in the immune system, is there anything that it can tell us about vascular dementia?
Yeah, I don't know very much about vascular dementia myself. I know that it's got quite a fluctuating disease course, and it has a lot more to do with kind of what's going on in terms of hypertension outside of the brain. I'm not a great expert on that. I know that there's inflammation involved in vascular problems, so high blood pressure and all those sorts of things increase your kind of the inflammation in the rest of your body, and then that has a knock-on effect for your brain. But I don't. Can you speak to that any more, Casey? Because I can't.
I think, yeah, it's we know we're beginning to learn. I think researchers across the country and across the world are learning more about the immune system and how it responds to these different changes in the brain. And I think in vascular dementia it's slightly different because you don't have this protein buildup in the way that you do in Alzheimer's, and that's where your kind of focus on is on that protein buildup. So yes, just looking at another question.
So are there any known associations between pressure on the brain head in scuba diving and Alzheimer's disease? They're interesting question. Leonie's got loads of interesting questions. That one I don't know about specifically in scuba diving, but there are lots of things coming through about brain injury. So things like rugby, where there's lots of high impact injuries to the head, they can trigger types of dementia, though I'm not sure that it's Alzheimer's disease, but it can cause an injury that causes inflammation, and that can increase the risk of dementia. With scuba diving, it would be really interesting because you'd have the pressure aspect, and you'd also probably have if there were a problem during the scuba diving, you could potentially have hypoxia, so a lack of oxygen getting to the brain, and that would also be inflammatory. So good question, don't know.
Yeah, it might be another one that we have to follow up after the event. But yeah, if people do have questions we're not able to answer live today, then do get in touch and we can try and point you in the right direction. I think I'll just hold on for another minute or two, see if anybody else has any more questions. But it's been really fascinating to hear about your research, Megan, and I just want to say a happy birthday to Grandma Joan today.
Yeah, I'll pass that on. How does dementia affect speech? That is a difficult one, and it depends very much on the type of dementia that you have. Some different dementias, as well as having different types of proteins involved, they affect different parts of the brain. So there are some that affect speech, and I can't think which one. I'm not a clinician, so I'm not very good at knowing which one's which. Do you need to translate that, Rachel? Are you happy to do that?
Yeah, so diolch am y cwestiwn. Mae'n dibynnu ar pa fath o dementia mae'r person yna yn cael, a mae'n dibynnu pa sort of pa adalau yn y menydd sy'n cael ei effeithio. So Megan, sorry.
Say that again, sorry.
Sorry, yeah, you can carry on.
Okay, cool. How closely are the researchers working with pharma companies to develop new drugs to treat dementia? It's really dependent on the different groups. I work in a group, my boss is called Paul Morgan, and he's this like genius guy who's been working in a field of kind of inflammation research for so long, years, since before I was born. And he has some really close ties with various pharmaceutical companies, and we're trying to develop treatments that slow down the inflammation in the body and might have an effect for Alzheimer's disease. So quite a lot of people are working really closely, and as part of the Dementia Research Institute, we work with DP UK, Dementia Platforms UK, and all sorts of different Oxford Drug Development, Drug Discovery, is ODD Drug Discovery, all sorts of different platforms to try and develop drugs. So yeah, we've got quite a few close collaborations.
Wonderful. Well, I think we've got one more, hold on. So this is a question about the immune system, so maybe you might have some insight here, Megan. If you have an autoimmune condition, does that put you at a higher risk for dementia? Do you have any insight into that?
So for other people who don't know, autoimmune conditions are conditions where your immune system recognizes something in your body and thinks that it doesn't belong there, and your immune system starts attacking itself. And I'm not actually sure whether that specific type of immune condition is associated, but I do know that lots of diseases that have increased inflammation outside of the brain, just within your the rest of your body, if you have increased inflammation, then that can increase your risk. But that does absolutely not mean that if you have an autoimmune disease, you will get Alzheimer's disease or any type of dementia. It's just like a subtly increased risk.
Thank you. Yeah, I think one of the interesting things, I guess, about research into the immune system is that it only really became a big thing in the dementia field in the last 10 years or so, with some key discoveries about genes about 10 years ago. So we're still, I guess, at the early stages of answering some of these questions. And so there's more research going on at the moment that we're able to talk about today. So okay, one more question, another question come through. I guess quite a topical question, is COVID likely to have an impact on these diseases? So I guess we mean neurodegenerative diseases.
Yeah, and I don't know anyone who's working on this at the moment personally, but I know that lots of people are. And so in my previous job, where I said about how my gran used to get infections and that would make her dementia worse, the problem with those sorts of studies is that it's difficult to capture the people who've had an infection. You don't necessarily know that they've had a urinary infection or not. Whereas with COVID, there's going to be the biggest cohort ever of people who have been infected. And often with these sorts of infections, it could be something that happens before they've got symptoms of dementia. So it's the sort of thing where people could sign up for studies now, say I've had COVID or I haven't had COVID, and then we could do longitudinal studies, so follow-up studies with them, and then find out whether it's affected their risk of dementia. And I suspect that it probably will, because there's an immune response, but how your body handles that will affect your dementia risk. Because if you have an infection and then it's resolved and you're totally fine, then great, that kind of suggests that your immune system is in good working order. So that might not increase your risk. But if you've had an infection and it's been really very serious, then that could potentially increase your risk. So it'll be really interesting to see what happens in years to come.
Yeah. Great. So then I think we can probably wrap up now. We've had a really good session, heard 2 really good talks. So I'd like to thank both Megan and Rebecca for sharing their work so passionately with us. And so I want to tell you about the next event in this series that we're planning. So it will take place next month on Tuesday, the 30th of March at 2 p.m. again, and it will feature the Brains for Dementia Research Centre in Manchester. Because you've come today, you won't need to sign up again to receive the joining instructions, as we will send those out to you a couple of days ahead of that event. We've put information about that one on the website as well, so if you want to find out more, do check out the website. And I'd like to thank you all from coming today and for your support for Brains for Dementia Research. As you've heard, because of your involvement with this study, we've got huge amounts of data and information that is really supporting our research. And so, we want to thank you for your ongoing contribution to the study. I hope that you're keeping safe and as well as possible in the current situation, and that you can join us again for other events and situation in the series. Thank you and goodbye.